friends. So in this video again I came with one more interesting topic that was from the heredity lesson. It is monohybrid cross. The Mendel, he has to be explained only one character, how it has to be passing from parent to progeny in this cross. The name monohybrid cross, the name itself, it has to be explaining that. So in that, mono means single. So here only one character he is observed. Okay, and hybrid cross, in the sense he has to be taking two pure breeds and he is going for a cross pollination. So like this he is creating a hybrid. So that's why it is calling it as monohybrid cross. So how we have to write in the exam, so I'll be explaining in this video. Okay, so first of all, in this monohybrid cross, so first we have to write a character. So here I am taking the character, it is a stem length. So tall and dwarf. So these are the two defined characters regarding to the stem length. Okay, so tall is a dominant character and dwarf is a recessive character. Okay, so now I move on to the monohybrid cross. So first parent phenotypes. We have to write the parent phenotypes. So what is mean by a phenotype? The physical expression of a character, we are calling it as a phenotype. So either how it will be appears, either tall, either dwarf. So the physical expression is calling it as a phenotype. Here the two characters are tall and dwarf. So next the genotypes. So here we are writing a genotypes as capital T and small t. So why? Because they are the pure breeds. So what is this genotype? A character is expressed in the form of alleles. What is mean by allele? The alternative forms of genes are called as alleles. Instead of the entire genome length, we are writing with a single letter. Okay, so they are called as allele. Each and every character, it will be having two alleles. So which will be two genes. Okay, two alleles. One from the maternal and one from the paternal. Okay, so it means that every character, it will be having two genes. Mendel, he called the genes as factors. Here, what are the genotypes? Capital T and small t. Okay, so next, what we are doing? Hybrid cross. We are going for a cross pollination of these two pure breeds. Okay, so what will happen? The cross pollination is we are performing the cross pollination. Why? Because pea plant, it has to be going for self pollination. Okay, so that's why here we are performing a cross pollination by doing a emasculation process. In emasculation, we are removing the anther from a flower, and with the help of another flower anther, we are transferring the pollen grains to that particular carpel. Okay, like this we are performing the cross pollination in between of these two pure breeds. So they'll be having pure breeds. That's why a single type of gene, it has to be present. So in the gametes, only a single set of genes, it has to be coming. So that's why in gametes, only one allele, it has to be present. So here two alleles are the homozygous. Homozygous means it will be having a same type. So capital T. So two are same type of alleles. That's why we are calling it as homozygous. Okay, so here the gametes, it will be having a capital T and small t. So tall, from the tall, the gamete is capital T. From the dwarf, the gamete is small t. When you are going for cross pollination, so whatever the resultant is capital T, small t, which will be a heterogeneous. So it means that the two alleles are not the same. So this condition we are calling it as heterozygous condition. Okay, so in this whatever the seeds it has to be formed, whatever the stem length it has to be appeared in the first generation. So that will be calling it as a F1 plants. First filial generation. First filial generation, which is also called as F1 plants. Okay, so these F1 plants are in heterozygous condition. F1 plants are in heterozygous condition. So here all are coming as a tall plants. Why? Because if a character is expressed in a heterozygous condition, one allele, it has to be dominant on another. So it means that only one factor gene, one factor physical expression is appearing. It means that so they will be dominant always. So whatever the characters are expressing in the F1 generation, they are calling it as dominant characters. The characters which are expressed in the F1 generation, they are calling it as dominant characters. Okay. So then next we are the experiment. It is not completed up to here. Okay. So then what they did, so how if when we are going for pure bits, it will be getting a heterogeneous condition. So how these two characters, it has to pass to the next generation. It means that we have to be perform the other experiments. So now what we have to do that, we have to go for self-pollination. So within the hybrids, whatever we get that in the F1, so with that we have to go for the self-pollination. So next what we have to do that, so self-pollination in the F1 plants. Okay, so F1 plants in the sense always in the heterozygous condition. So the F1 gametes, what are the F1 gametes? It is a heterozygous condition. So here we have to be apply the law of segregation. So the two gametes, it can be able to segregate independently. It can able to segregate and it can able to fuse with any other allele. So it will be having that capacity. So that is called the law of segregation. Again, I will explain the laws in the last. Okay. So here the two heterogeneous condition, how many different types of alleles are present, that many gametes it has to be formed. So here two different alleles, heterogeneous condition. No. So capital T, small t. So capital T is one gamete and small t is another gamete. So F1 plants, same type of plants. So here also capital T and small t are the gametes. Okay. Now, so if these two gametes, it has to be going to fuse and forms the F2 generation, second filial generation. In the second filial generation, we can write like this. So we can cross it like this. We can fuse like this here itself. So this capital T, it can able to fuse with capital T. So then it has to result and form that small t, capital T, capital T. So if this capital T can able to fuse with this small t, so at that condition, it has to be formed as a capital T, small t, heterozygous condition. The same thing it has to be applicable for this small t also. So this small t, it can able to fuse with the capital T. So at that time, it has to form a capital T, small t. And this small t, it has to be fused with the small t. And the resultant, it has to be small t, small t. If a monohybrid cross, we can able to fuse it here itself. But if we are going for dihybrid cross, if we are going for trihybrid cross, at that condition, so this type of fusing is not at all possible and it will be very confusable. So at that time, we have to go for a Punnett square board. We have to go with the Punnett square board. So this Punnett square board is commonly calling it as a checker board. Okay. So in this checker board, we have to be drawn. So how many gametes are there? That many columns and that many rows. And one more row and one more column we can draw that for placing the gametes. Okay, here see that. So in this row we have to be writing one particular plant gametes, capital T and small t. Here one particular gametes like capital T and small t. So if you see that, so here capital T, if you fuse these two, capital T, if you fuse these two, capital T, small t. Here if you are fusing this capital T, small t and this 2 it has to be formed small t and small t. Okay, like this we are getting a F2 generation. So from this F2 generation, we can write the ratios. So we can write the monohybrid cross phenotypic ratio as monohybrid cross genotypic ratio. So the phenotypic ratio, what are the two phenotypes, tall and dwarf? So how many tall? It has to be appeared 3 tall. So then 1 dwarf. Then the ratio is 3:1. The monohybrid phenotypic ratio is 3:1. So then monohybrid genotypic ratio. So here see this, the all tall is not in a homozygous. So 1 tall it has to be in homozygous and 2 tall it has to be in heterozygous condition, and 1 dwarf. Dwarf always in the homozygous condition. Why? Because recessive it can express only when the 2 factors are belongs to the same. Okay. So here, so 1 homogeneous tall and 2 heterogeneous tall and 1 homozygous dwarf. So the genotypic ratio is 1:2:1. So monohybrid cross genotypic ratio is 1:2:1. So from this monohybrid cross, Mendel proposed 2 laws. One is law of dominance, another one is law of segregation. So now I'll explain about the law of dominance. So first we'll see about the law of dominance. So each and every factor, it has to be expressed by 2 alleles. 2 alleles. Okay, either 2 factors. Okay. So then if you are seeing that when these 2 factors are in heterozygous condition, if it will be in homozygous, there is no issue for that. So either capital T in the sense, there will be tall and small t in the sense dwarf. So when it has to be in the heterozygous condition, at that time, so one allele, it has to show the dominant on another allele. So this is known as law of dominance. For example, so capital T, small t, it is in heterozygous condition. So in that, the 2 alleles are present. One allele is for tall, one allele is represents the dwarf. 2 alleles are present. Even though one allele, it has to show the dominant on other. So the capital T, it has to show the dominant on small t. So this is known as law of dominance. So when a character is expressed in a heterozygous condition, so one allele, it has to show the dominance on another. So this is called as law of dominance, which is proposed from this monohybrid cross. Okay, the another law is law of segregation. The segregation means separation. So generally, if a character is expressed by 2 alleles, we all know that. Okay, so the 2 alleles, it has to coming from maternal and paternal, isn't it? So when at the time of gamete formation, so these 2 alleles, they can able to segregate individually, they can able to separate, and each and every allele, it can able to fuse with any other allele. So this segregation, this separation, we are calling it as law of segregation. So see this, what will happen? A pair of alleles for a trait, and that each parent randomly they can able to separate. So generally, at what time it has to be appears in the sense at the time of gamete formation. Okay, for example, so capital T, small t. So it is a heterozygous tall. We all know that. In that, so capital T, it has to came from the tall, and small t, it has to came from the dwarf. Okay, so here when this F1, it has to going for gamete formation, the capital T, it can able to separate. It can forms a separate gamete, and small t, it can able to form a separate gamete. So this type of segregation, this type of separation, we are calling it as a law of segregation. So this law of segregation explains that when at the time of gamete formation, the alleles they can able to separate individually, and they can able to fuse with any other alleles. So this explains about the law of segregation. Hope you people are understood this video. If you like the video, please like, share and subscribe. Thank you for watching.